There was originally a simple and widely accepted argument that the genetic code should be universal, namely, that any variation in the genetic code would be lethal to the organism. This is known as the frozen excident argument for the universality of the genetic code. However, in a seminal paper on the origins of the genetic code in 1968, Francis Crick still stated that the universality of the genetic code in all organisms was an unproven assumption, and was probably not true in some instances. He predicted that the code is universal or nearly so. The first variation was discovered in 1979, by researchers studying human mitochondrial genes. Many slight variants were discovered thereafter, including various alternative mitochondrial codes. These minor variants, for example, involve translation of the codon UGA as tryptophan in Mycoplasma species, and translation of CUG as a serine rather than leucine in yeasts of the CTG clade. Because viruses must use the same genetic code as their hosts, modifications to the standard genetic code could interfere with viral protein synthesis or functioning. However, viruses such as totaviruses have adapted to the host's genetic code modification. In bacteria and archaea, GUG and UUG are common start codons. In rare cases, certain proteins may use alternative start codons. Surprisingly, variations in the interpretation of the genetic code exist also in human nuclear encoded genes. In 2016, researchers studying the translation of malate dehydrogenase found that in about 4% of the mRNAs encoding this enzyme, the stop codon is naturally used to encode the amino acids tryptophan and arginine. This type of recoding is induced by a high read-through stop codon context, and it is referred to as functional translational read-through. Despite these differences, all known naturally occurring codes are very similar. The coding mechanism is the same for all organisms: 3 base codons, tRNA, ribosomes, single direction reading and translating single codons into single amino acids. The most extreme variations occur in certain ciliates where the meaning of stop codons depends on their position within mRNA. When close to the 3' they act as terminators, while in internal positions they either code for amino acids as in condylostomum magnum or trigger ribosomal frameshifting as in euploatase. The origins and variation of the genetic code, including the mechanisms behind the evolvability of the genetic code, have been widely studied, and some studies have been done experimentally evolving the genetic code of some organisms.